Hello, everybody. And some kind of points before I even start. As you've seen earlier, both in ChIP-Seq and in Seq, the surveys from last year were very helpful for us to improve the session for this year. So I encourage each and every one of you to write what they liked, write what they didn't like, and give us some future suggestions, because we believe in educating as many people as possible and making it available to everybody. The other point is, like, as I mentioned earlier, and you'll see it with the modules that I'm going to present, the idea of the hands-on is to give you kind of a feeling of how would you address raw reads and how would you analyze them, and also to expose you to some of the tools that are coming more and more available nowadays, such as gene pattern. And on this regard, also, suggestions can be very helpful, because we've been working, and in my acknowledgement slide, there's a lot of people from Gene Pattern and Jim Robinson from IGV and his group, and they're very responsive to suggestions and thoughts. So again, feel free to email me, and I can relay you to their groups. They have like kind of Google groups in the same way that I've set up for all of you. And any ideas and points to improve will be terrific. And I can tell you that some of the modules that we're using are, especially for the chip sequencing, are in the building. So they're just, again, to give you a taste. But the idea is that in the near-ish future, you will be able to use them to analyze your data. And let's see. Yeah, so I'll just go over some kind of a brief introduction, and I wouldn't repeat too many of the points that Chuck and Noam presented, but again, I'll be happy to address any questions, either in front of everybody or in person. And my major goal here is to give you a feeling of how to do hands-on analysis, because I did my PhD in experimental biology, and during my postdoc, I invested a lot of time and effort into getting some idea of how to address big data sets, such as chip sequencing. And what I love and what happens today is that some of these efforts are overcome by tools such as gene pattern. So my point here is to show you how the data kind of looks like, and to give you an idea of how is it built, and some kind of a feeling of how to use it. And I hope that you will get that from the hands-on analysis that we've built. So you all heard about, where is the pointer? Okay, you all heard about chromatin and the way it's organized, and about ChIP. And I also saw that Noam had a slide. I think that the bottom line that I want you to get here is that when you, so let me, before I even go there, how many of you here are experimentalists that have never touched Unix or any other computational environment? Don't be ashamed. Okay? And how many of you have ever, like on the other end, how many of you have ever done some kind of analysis? So I'll see that people do answer. Okay, so there's like exposure from both directions. And in general, the idea is that when you take your library and you put it on the sequencer, it spits out a file that has a lot of reads, which are just sequences of letters. And then you need to align them to a genome of reference, which is one of the things that we're going to do today. We build kind of maps of like where do they align and where is the highest levels, and then we get the peaks that you've seen. And the hope today is that that's something that you're going to do, kind of imagining that you get the reads from some public data or from a machine, run it through the pipelines, which to an extent are black box still, but less of a black box, because you at least see the procedures. And then I want you to see on your own machine how does it look from beginning to an end. I'm not going to go too much into this, but some of the ideas that we can talk about are the chromatin regulators that regulate the genome. And if you're interested, there is a nice website of some work that we've done on that. There is the ENCODE consortium studies that they have a lot of connection, and they even have like a nice iPad application. And as Noam said, one of the things that was really exciting for me, when I was a graduate student, I think we knew about, I don't know, 5, 10, 20 different enhancers. And most of them were discovered by kind of deletions. And nowadays, people are talking on the orders of 100 to hundreds of thousands different enhancers. And this is one of the beautiful power of ChIP sequencing, that it allows us to identify and annotate genomic regions that were not amenable for this kind of identification before. That's again giving you some of the studies that were looking into enhancers and how do they bind and what is the exact motif that is identified, as Noam said, by a specific transcription factor. So before I go on to the workshop, which I will go with you over kind of from a bird's eye view, and then I want each and every one of you to try it. I want to acknowledge all the people that helped with building up the script and the code for that. So the ICM group, a group of people that were working with me on analysis of a project that I'm interested in, which is early development, and all the gene pattern and the IGV people that were engaged in our excitement of sharing these tools with the community and implemented them into the gene pattern. So in general, I want to start with the hands-on, and I will bring in the let's see if that works. Okay, so I want you all to try and follow up the PDF that I've sent you. How many of you have logged into gene pattern and have tried it? Okay, that's a good number. And in general, what I want you to do is as follows. So many studies, or nowadays, whenever we do a study, either for ChIP sequencing or RNA sequencing or any genomic study, the beautiful part is that we can deposit the data into public domains, such as Geo or ENCODE has all of the data in specific locations and so on. And what makes it really nice in my mind is that now you can follow up on other people's study and use what they've done in order to further the knowledge in biology. So what I want you to do is imagine that you take row reads from data, and what I've done is exactly that. Like, if you follow the instructions and directions, instead of giving you an entire human genome, which will just take a bit longer, I've reduced it into one chromosome, which means that I had to align it first because the reads are not aligned, so you don't know where they are. So I aligned it to the genome, I took the chromosome 19 only, and then I unaligned it. So now it's kind of a non-aligned, only chromosome 19. And the idea is that it comes as a Fastq file. Fastq and all these names are things that you can Google and Wikipedia. And I tried to send you the link to the SAM tools. But most of the machines are providing Fastq files. And then you align them to a reference genome. And you get what is called a BAM file, which is the binary of SAM files. And again, this is part of the kind of basic aligners. And one of the key people that built it is Hang Lee, which actually works here at the BroadE. And it's very fun to see him wandering around and see how much he has implemented and change the field of alignment. So what we're going to do is we're going to take Fastq, which is what you get out of a sequencer. We're going to align them into the genome of reference. So I took ChIP sequencing data for H3K4ME3 and H3K27ME3, and a control, which is the whole cell extract, which gives us kind of a reference of how would like the non-enriched, non-fractionated genome is. And then we will convert them into bumps, use IGV to visualize them, and then we can do some further analysis. But this is kind of an overview of the workshop. And before we dive in, I want you to ask me any questions, and then whoever feels that they are comfortable doing it more in Unix, which is much quicker and much more flexible, can either stay or leave. Again, I just don't want to take anybody's time. Any questions before we start? Okay, so the way I usually run this hands-on analysis is that you all tried. I hope that some of you have tried it or at least touched it before. And if something doesn't work or if you have any issues, let me know and I'll try to solve them. If not, I will try to show you what it should look like. So. If there are questions, I can also, try to come specifically to whoever has it. Trying to make it easy on everybody, the way we build it is that if you go to each one of the modules, so let's start with the first one. So I think it's going to save some time. So I go to Homo sapiens HG19. We don't need a custom BroadE index. The input format is Fastq. And then the read pair one, you click on add path or URL, and we've already put the files for you there. So you go to raw data, and then you can just drag and drop it. So I drag it to here, one. But before I can drag more than one, I tell it to do a batch. So it's going to run on all three of them at once. 2 and 3. And then let's see if it needs anything else. It doesn't need anything else. I just do cancel on this, and I click on run. And then it just submits the job. When it submits a job, it means that there is somewhere a big, strong, fast machine that is running the job for you. And now they're running. So sometimes it takes like 5 minutes. Sometimes it takes shorter. I think it might take a bit longer because I guess all of you are going to do it now. Yeah.
So I have like.
So one point that I realized was not clear enough, and we'll see it later when you get to the IGV, is the idea of the whole cell extract or control. And let me go for a second to the presentation. Okay, so the basic idea of ChIP, if we really zoom out and think about it, is that what we do is we actually fractionate the genome. So we took cells, we cross-linked the proteins with the DNA, and then we chopped it into small pieces or sheared it into small pieces, and we used an antibody that recognizes the epitope that we care about. So now we've pooled the pieces of genome or the pieces of DNA that were in association with this protein from the rest. So it's an enrichment or fractionation procedure. Our control for that is to take exactly the same cells, shear them in the same way, but not use any antibody at all. Just take them the way they are. And we call this whole cell extract or control. And the idea is that because there is dependency on the way the chromatin is organized in the cell, or in the way we shear the cells, or in the way we cross-linked it, and so on, we want to do this control for each and every experiment. But if I take cells, and then I use like 6 or 7 epitopes, like for H3K4ME3, H3K27ME3, CTCF, and so on, one control, if I started from the same population and I just split it, is enough, because it gives us a good enough reference for all of them. But if I want to compare different conditions, I would want for every time, every batch of cross-linked and biological material, I would want the whole cell extract, because the conditions differ. So I want as good of a reference point as I can. And the idea is that the control should not be enriched. In general, we do see biases even in the control, and I think that that's one of the things that you can do using the data that I gave you, because it does have a control. So since open chromatin is more sensitive for shearing, if you look on, for instance, on the whole cell extract, there's more enrichment for reads over the transcription start site, over the promoters, because it's more of an open chromatin. So we do get some kind of a bias, and it's good to know it. So you can keep on going. I realized that this was something that was not clear. How many of you have gotten to the point that they have BAM files? Okay, good. So it seems that we're moving pretty quickly. So one thing that you can do, let me go down to, where is it? Okay, so let's imagine, do you see this? Yeah. So if I click on it, so if you click on the BAM, you see that it opens up kind of a different options to do with it. And one of them is IGV. So if you click on IGV, it says launches IGV with the input files. So I click on it. And in general, this is more relevant to RNA sequencing, but it's also nice to do it with ChIP sequencing. It just takes a bit longer here. Let's see. Okay, I'll just do it. Keep on doing what you're doing, and I'll see if it works quickly enough, or not. I'll give you another option to do it.
So if you open IGV independently, do you then have to save the output files locally? Or
Yes. Yes. The gene pattern are working on implementing it that you can run everything connected, but it's only in the production mode. It hasn't gotten to the public one. But what we can do is
you shouldn't use the TDF. So we just forgot to go.
So if I just drag the BAM to the IGV, in general, you wouldn't see anything, but if I go to chromosome 19, okay, and zoom enough, okay, you can really start seeing the reads. So this is after the alignment. So you can really see where the reads are. So for instance, see all these areas have a lot of reads aligned to them. But this is not such a comfortable way of viewing it. For instance, this is a really good area. You see like it has several reads aligning to it. The TDF, which you will generate soon, is a better way to view it. So, but I also wanted you to see how does the raw reads look like on the IGV, which I think is really nice. It gives you the directions and so on.
So what should be the maximum number of lines in terms of having a good data?
This depends on your data and so on, but that's a good question. It depends on, is it ChIP sequencing for which epitope, if it's RNA sequencing and so on, what's the size of the genome of the model organism you're working with. It varies from, bacteria or viruses are nothing, and humans or even more complicated genomes are so, but it depends. Okay, I think this is actually something that for me was really exciting to see that, like, if you look at the reads at the beginning, which I'll show you in a second how they look like, they just like, it's starting from reads and then really visualizing them. So what I want to do is I want to show you how I made them into TDFs. So for instance, I'll do it for one, but in general it's the same. And it's, again, now we have to do it manually and we have to download it into your machine, which is good that I've made it only into one chromosome. But in general, the future will be that you can do it still in gene pattern and connect it directly to IGV. So if I go, for instance, to the file that we've generated, which is the BAM for A3K for ME3, you see it here, I hope. I click on it, and one option is to save file. So let's say that I make a new folder in IGV. I called it BroadE 2014. Okay, and then I save it there. The next step will be to go to IGV. And once I've opened IGV, there is something that's called tools. I go to Run IGV Tools. And from Run IGV Tools, it asks me to browse. So I browse the one that I've just generated, which is in here. I click on it. And it's going to name the TDF file. And the same name, it's just going to add TDF at the end. And I run. Since it's only one chromosome, it takes pretty quickly. Usually it takes a bit longer. And now, if I go to it, I can just drag it. So I see the TDF and I see the BAM. I can just drag the TDF to the IGV, and it's here. So we're going to have duplicates because I've already did it before, but this is the one that we've just made. And so and again, just a matter of which resolution you do it, but this one and this one are identical. It's just that I've capped one of them in a different size. Again, the idea is that we took raw reads from the server, we aligned them to the genome, we made some file which is a file that we can read, which I'll show you in a second how it looks like on the Unix server, and then we've made BAM files that are easily for the machines to use, and then we used one of the tools in IGV to make it something that you very easily visualized. We also saw that we can visualize the BAM file, but for instance, the TDF includes some extension of the reads into directions and so on. But I want to show you to demonstrate some of the features of IGV. I just want at least most of you to get to this point. And this is, again, what I was doing. I downloaded the BAM. I went into Tools. I did Run IGV Tools. And then in Run IGV Tools, from Browse, I went into the directory where I placed the files that we downloaded from Gene Pattern. I just double-clicked on the BAM file. And then I did Run. It's going to ask me if it's overwrite. I just say, yes, it doesn't matter. For you, I hope you don't need to overwrite that. You just do it once for each of the files. And then we should have 3 files, A3K for ME3, A3K27ME3, and the control. Once you've got to that point, I want to show you how to call them differently and how to make them in the same size and how to view them in a comfortable way in IGV. Okay, I want to give you, I will revisit it later with people that didn't get to the IGV yet, but I want to give you some feeling of how it works. I use like a lot of right-click, and for instance, if I right-click on this, I can do remove track because I put 2 of them together. And then you have this one here, which tells you resize tracks to fit into window. So that's nice because it fits it into the window. Now, if I use Shift and click on all of them, and then I do, right click, I can do set data range. Now, because we want to compare tracks between different samples, I want to make sure that all of them are on the same data range. Sometimes it makes sense, sometimes it doesn't. In general, you also want to make sure that the read depth is the same. But again, this is where the field of epigenomics is still trying to find the best ways to normalize. But for our case, let's imagine that something like 15 is a good maximum for setting the threshold. So it's a matter of signal to noise. Now, one really nice thing that you can see is you have home here. Home means go to the entire genome view. So you see this is the entire genome, and you can see that we have kind of a blip over chromosome 19 because our original file was only from chromosome 19. So if I go to chromosome 10, for instance, it should have nothing apart from just regions that are repetitive and random and have actually nothing in them. But if I go back to the home and then I go to chromosome 19, we can see the 3 tracks. Now, I want to make it nicer on my eyes. So the convention that we got to is that the color that we like for active marks is more towards the green. So I'm going to choose some kind of green for the K4ME3. The one for K27ME3, which is more towards the repressive, is more on the reds end. So I'm going to do that. And the control, many times we just make it as black. But again, it's totally random. You can do whatever you feel like. Another point that someone asked me is how do you save figures? So if you just want to show someone a figure of how it looks like, I use screenshots, which in my Macintosh I made a shortcut for it, but I'm sure that you can figure it out, just Google screenshot, and that's very easy. In terms of the IGV itself, so you have save image. Now in save image, let's just go to here, for instance, and do downloads. And okay, so you have PNG and you have JPEG, but you also have SVG. And the beautiful part of SVG is that then you can open it with Illustrator and modify it. So, and in general, not only IGV, any web tool that you use and creates images for you, it's great that they would make SVG for you, because SVG is Adobe Illustrator friendly, and then you can make it look really nice and create nice PDFs out of it. And then you save it, and then you can open it in Illustrator. Another point that I really like is you can zoom in on, I select an area and I zoom in. You can also define an area. So for instance, if I want to zoom out, I click together with Alt. You can also use these, of course, the plus and minus, but clicking Alt makes you zoom out. And then imagine that I want to decide what's the size of this peak. So you can click on this one, which tells you define a region of interest. I click on one side of it. I click on this first. I see it clicked in. I click on one side of it, and then I click on the other, and then I get this red thing. Now, what's really beautiful about it, as you can see, it tells you exactly the coordinates. So it's chromosome 19, 399, 70, 730, and so on. The other thing is that if I double click on it, I can do zoom on it. I can copy the sequence to my clipboard. I can edit the description, or I can delete the red line. So this is a nice feature of IGV. And again, I can talk forever on IGV. I want you to do some more stuff. But look into the IGV. I found IGV one of the best tools to view the genome and do points in it. One last thing that I will show you is the regions option. So for instance, you can select genes. I'm not sure if it's going to work because we have only chromosome 19 here, but let's imagine that some of them will be. And then let's do view. Okay. And then you can see many, many areas at once. So you see some of them are chromosome 19, so we do see peaks on the promoters of these genes, and some of them are not, and that's why we don't see anything. But it's really nice to see that we can see many, many genes at once, and you can define it and so on. And again, I totally encourage you to go into the instructions and the interface of IGV to explore it more. So I know that some of you haven't gotten to the IGV point, but some of you did, so I was trying to go with the crowd. But if any of you has any more questions about it, I'm here. Okay, so I want to show you something. So if I go to Unix and I look at the original Fastq files, that's how they look like. I can enlarge it. This is like one of the Fastq, which, by the end of the day, these are just reads. So what I want you to appreciate is that we started from this point where we had a file that looks like that and can go on forever because it's pretty big, even though it's only one chromosome. And then we aligned it to the genome, and now I think most of you, as much as I've seen, managed to view it on IGV. So I think that's already a really good point. In like 5 or 10 minutes, I want to go to the next step, which I think I call it section 2 or something like that in your workshop. But the idea is that we want to take it and look at many regions at once, kind of using other visualization tools. So apart from IGV, which is kind of Google Maps for visualization of specific genes, and to some extent, a bunch of genes, there are many ways, for instance, where you want to look at 70,000 different enhancers or 10,000 different promoters at once. And this is more or less where I want to take you to get a feeling of what is the feasibility on this regard. So I'll try to give, like many of you, the time to get to this point. But if you didn't, let's try to get to the next step where at least all of you make sure that you have the 3 BOM files. So if there's someone here that didn't get to the point that they have the BOM files, the aligned BOM files, let me know so we'll get to that point. Okay, I see that some of you already started the next step, which is great. But there are some bugs or points to fix in the way it's built that I think will make it easier on you. So first of all, in the extract signal, you do need to use the batch where you use all of them. But in the next step, even though you have the batch option, don't use batch because it actually requires multiple files. We'll get to it in a second, but just remember that. The other point, which I think is really important, so the region bed file, some of you, not sure where it comes from, because I made sure that we have only for chromosome 19. Some of you gotten, I don't know, an old version. Perhaps it's in the storage that I put some old stuff, but make sure you use the one that starts chromosome 19 TSS table.bed, which means that this is the TSS, the transcription start site table for chromosome 19 only, and bed, it's a name of a file which tells you chromosome, start point, end point. And you can add some other features, but that's the definition of a bed file. So this is the one that you should use, and you shouldn't get bin files for all the chromosomes. If you get that, it probably means that you've used a wrong file for the region bed. So these are the 2 points that are kind of bugs that I've seen repeating for some of the people. When you do the create heat map, make sure that you don't use the batch. Also in create plots, you don't need the batch option. You can use multiple files here. And when you use the bed file, make sure that you use only the chromosome 19 one. In general, when you do it for your files, which are genomic, you want all chromosomes. But for our exercise here, we just want chromosome 19, so otherwise it will take much longer. And whoever is leaving, please make sure that you fill up the forms either today or tomorrow, whenever you want. But just make sure you fill them up for the future. Okay, so I see that some of you have already started the next step. I just more or less want you to get an idea of what we're doing here. And one of the things that we try to do is also to give kind of a short description. So for instance, ChIP-Seq extract signal, it says that it's meant to read from a BIOM file into bins by chromosome and extract the signal values. And the idea is that in general, what we're trying to do is to instead, it's somewhat similar to what you do in IGV. And what we're doing is that we start by taking the entire chromosome. So this script should run on all chromosomes. And this is why it requires the sizes of the chromosomes, because different organisms have different lengths of chromosomes. So again, we have only one chromosome, so it doesn't really matter in our files. But it goes all over the chromosome and breaks it into 25 base pair bins. And then it takes all the reads that we have in the BAM file, and the BAM file tells you for each read where is it located on the genome. So it takes all the reads and asks, okay, for each of these 25 base pairs bins, how many reads were touching it? So instead of taking the chromosome to the, like, I can just flip the chromosome now. So let's say this is the beginning and this is the end of the chromosome. And again, we have many, 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 many bins here. And let's say that we have a gene here. And that's its promoter, or the way we use it here is the TSS, the transcription start site. And for promoter, the regular convention is that we take approximately 2.5 kb upstream and downstream from the transcription start site. And so we have 25 base pair beans, and then we do 2.5 kb up and down. So this means that we have 100 beans in this direction and 100 beans in this direction. And let's say that most of the signal is going to be here. Let's say that I don't want to start at the beginning of the chromosome. And here we don't have a lot of signal. I think you've seen it pretty nice when we were doing in the IGV. Okay, so here, for instance, is a gene, and this gene goes to the left direction. You see that most of the reads are falling here. Now, instead of just like, the first step is to take the chromosome and bin it into 25 base pairs. And then we take the reads that are on the BAM file, and we ask for each of the bins how many reads we're touching it. So if the reads are 100 base pairs, they can touch many of the bins, but it doesn't matter because we care about each and every bin. And then the next step that we're going to do is to count for regions that we care about how many of these reads were aligned, and we also can do some kind of normalizations. But what's really nice about it is that using these normalizations and using this kind of conglomeration approach, we can look on thousands or tens of thousands of regions at once. So the way that it works is as follows. We start with extract signal, which it takes the BAM file that tells where the reads were aligned, which tells you for each genomic location how many reads were there. It takes the chromosome sizes file, because as we said, each organism has different sizes of chromosomes. It asks you if you want to normalize or not. It asks you what's the bin size. I told you the default that we made was 25 base pairs, but you can make it variable if you want. You can also make the region bin size different, which means that you conglomerate, you can merge certain sub-bins into a bit longer size, and you can even do an extension. Now, the extension is for what we call the region bed. A bed file, which is something that you might encounter, is just a way to define chromosomal area or genomic areas that you care about. And the way the most basic bed file will have, it's not going to have header, but it's going to have chromosome, start, and end. So in our case, it's always going to be chromosome 19. Start can be anywhere on the chromosome, and then end can be anywhere. And this is like, by the end of the day, it just tells the script or the code where to go and where do we measure these kind of reads, or where do we take the bins from and measure them and normalize. And this is like our first script. So we call it extract signal because by the end of the day, we try to extract the signal from the BAM file on the genomic areas that we care about. We don't want to do it for the entire genome. So we kind of look on the subset of the genome that we care about. And to make it easier on us, I took the promoters, which are the transcription start site plus minus 2.5 kbs. So the way I was doing it, and I will take you all through it because I saw that some of you had weird issues with that. Here we do a batch, so let's do it for all of them. And let's take, so we have these two BAM files that I put under BAM files. I also made some storage to make it easier on me, and I don't need to look for before. I have the control BAM. I even have a CTCF that I didn't want you to run it so it wouldn't take so long. I have K27, and I have K4ME3. So now I'm doing it for 6 files at once. The next step is we need chromosome sizes, which just make it easier on our eyes. We have HG19 chromosome sizes, so I drag it here. We also need the bed file. I make sure that I take only the chromosome 19 bed file.
What is bin size here? What is the upper bin size?
So a default is to take each and every of the chromosome and count how many reads were falling in consecutive bins of 25 base pairs. You can change it into whatever you want.
We didn't use this one. We used the one down below.
Yeah, because it's a different one. Yeah. So the regional one tells you how do you kind of disperse the data. So if you take bin size of 10 kb, they're not going to be so sensitive. So they're going to count everything. If you take 25, the resolution is pretty high. So this is like the way to imagine it. So 25 base pairs or 100 wouldn't make a lot of difference. But if you're taking 25 or 100 kb, then you don't have any resolution. And region bin size is when we look at it, when we visualize what's the size of bins that we want to use. 100 base pairs is a nice one, but again, you can. It's just a matter of resolution versus how big the file is going to be. And extend by, this means that it takes the center of the bed file and extends it in 2.5 kb in each direction. Now, since a file is the transcription start site, and so for prokaryotes, we really know what's exactly the promoter. For eukaryotes, it's not super clear, but the convention is something like the 5 kb around the TSS, around the transcription start site. So we take the transcription start site and we extend it 2.5 kb upstream and downstream. So this is why we extend it here in 2.5 kb. And you have functions that you can make, average or maximum or median, and then we just do run. And this is going to run. And the idea is that this is going to make us what we call tab files. Let's see if I can if we have a tab file somewhere. No, I don't have a tab file out of hand. Okay. While you're going to work on it, I'll try to find a tab file that we can look at, and I'll let you see how they look like, because in general it's also good to visualize files. Let's see if it's still running. This takes a bit longer because it kind of requires a bit more memory, because we have something like 1500 times 6 files, time extension of 2.5 KB in each direction. And while we're waiting for that, I will already do the next step, which is, I think create heat map is the one that we want. And if I go to jobs, let's see if I have it. Sorry? Yeah, here we don't use batches. And we have a much better version of the code for here. Trey Dixit that was working with me on it. We had a much better version, but we still didn't get to implement it into IGV. Okay, so now my jobs are done. Let's see the jobs. Okay, looks so good. I go back here. Okay, modules, and we say we want to create heat map. I take jobs, and I don't do batch, but I select all the tab files. So we should have 6 of them. Okay, and then I just click on run. It should be running. And I also wanted to download the tab files so we can look at it. Okay, so it might be easier to do it like this. Okay, so this is the tab file. It has chromosome, start and stop, and then adjacent start, adjacent stop. It again, these are like definition. But for each of the beans, which you can see, it goes for quite a long while. It gives you what's the score of how many reads are falling. So, for instance, in the first one there was almost nothing, and then you had 827, and then 70, and so on and so forth. And this is kind of a big file, but for each chromosomic location that we gave it in the bed file, for each promoter, it's going to tell you how many reads were over there, or how many beans, how many reads were touching the beans that we subtracted. Okay, and now let's see. Okay, so my jobs are done. Okay, so this is the job that I've ran. And now you can visualize, for instance, the... So there are many, many features here. What I want you to see... Where is the PDF? Oh, wait. There is an error. Okay. Okay, we might have gotten an error. Okay, I'll let you start with only 4 of them, because I see that I get an error when I use all of them, and I'm not sure what was wrong there. But let's do it again. No.
And the normalization is occurring just within each file? So
Yes.
You don't need to have all the files from the same experiment?
No, I mean, again, this depends. In general, we also implemented the way that it can normalize to the whole cell extract. So then you would want an whole cell extract for each experiment. But here we didn't.
If we add, if you were
noted
later, we add these other files for replicates, we don't have to normalize them in the same.
No. So this, I hope, conveys to you a bit the way the file is organized while we're waiting for the other stuff to run. So this is going to say chromosome 19. I've made it smaller because it goes on for a long while. But it's going to be chromosome 19, chromosome start, chromosome end. And then this is kind of some more points on the chromosome for allowing some kind of further analysis. And then it tells you what's the score or how many reads were falling on each of the bins that we were looking at. And it goes on for a lot of them because we said 2,500 in each direction. Okay, so this should be okay now. And okay, so what I've done now, let me just download it and then you can see it. I want to download it. Okay, so what you see here, the first one is the whole cell extract, which, as we expect, is mostly noise. The second one is K4ME3, if I'm not mistaken. And the third one is CTCF, which is one of the transcription factors. So in general, what's really nice about this, and one of the reasons that it was important for me to show you, is that it shows you approximately 1,600 or 1,650 promoters at once. And then it makes the visualization much more powerful because it's really hard to look at so many regions at once. So each line here is a genomic area, so you can stack one on the other many, many, many lines. At a certain point, even the resolution of visualization goes awry, but at least at these levels, it's really nice. And then the other point is that you can see this is the center, and over the center, you have high level of binding, and in the vicinity, it kind of goes down. And we also can see, like, if you look at it, and you can look at it on your computer, you can really see the 100 base pair bins. Because this bin has a bit lower signal than the one next to it, and you can see the differential. Now, if we will make the bins bigger, if we were making them 500 base pairs, then the resolution will be much lower, and the other way around. So I think this is one thing that is really kind of nice to see, is how we can take many genomic areas of interest, let it be promoters or enhancers, and take our data and explore it over it. Or if someone asked me, how do we compare technical or biological replicates? So it's nice to have biological replicates one next to the other, and this way you can look at many genomic areas and see how well do they reproduce on, like, for instance, all the promoters and so on.
I'm sorry, can you explain what's on the X and Y axis?
Sorry. Thanks for, let me also put it on the screen wide. So let's see if it works. So what you see here, this is the whole cell extracts. This is, I'm going to also say what you see in each one, but there's all cell extract on the left, CTCF in the middle, and K4ME3 on the right. Now, each line is exactly the same genomic area, which is the regions that we defined by the bed file. And the regions we defined in the bed file were all the transcription start sites, which you have approximately 1,600 genes on chromosome 19. So it's all the promoters of chromosome 19. And promoters we defined as the transcription start site plus 2.5 kb in each direction. So you have here the center. It's easier to see here. So here's the center that we defined. And then 2.5 kb upstream and 2.5 kb downstream. And the signal is the z-score, which means how much is each area enriched compared to the rest in each of the files.
Okay, so within each column, let's just say within the three.
So let's say we have here 3 columns.
The entire size of that is, we're talking about
5 kb.
So that's a 5 kb.
Exactly.
And then across chromosome 19, you're seeing different regions. So from the top to bottom within, or from the top, I'm not exactly sure.
So let's say it goes like, it just numbers how many regions you have. So it goes in 1,650. Yeah.
But so theoretically, base pair 0 would be somewhere.
This depends how you organize it. Here we are.
In this case.
In this case, it's not ordered according to the chromosome. It's ordered according to the signal. But it depends on, like, so it's not necessarily that this is where it is on the chromosome. It's more where is the highest enrichment of the signal. So we're trying to,
These have been K-means.
Yes, yes.
But that's the organization.
So we've made K-means clustering of that in order to, and within each block, the order is just according to the location. Sorry?
So how do you identify what gene you're looking at?
You can then later on go to the file and say, okay, which genes it was. But
So don't do a text file and
yeah, yeah. But it's one way to explore your data, to say, okay, I want to look at all 1,500 promoters. I want to look on all 150,000 enhancers. And then you can go back indeed and say, well, what's the genomic location within them? What you got is something completely different. I'll come and see what you got, and I'll try to recapitulate something similar.